Our speaker today is Dr. Procopio. Dr. Procopio is a faculty member in Dr. Heidi Jacobs' team at Harvard Medical School. He received his PhD degree at McGill University. He has published many papers on a variety of topics. Today, he's going to talk about some of his research, Locus coeruleus dysfunction in the earliest stages of Alzheimer's disease. Now, without further ado, please join me and welcome Dr. Procopio.
All right. Can you hear me well?
Yeah, perfectly.
And are you able to see my slides?
Yes.
You are. Okay, fantastic. Well, hello all. First of all, I wanted to thank Kelly and Dan for the invitation to present my work in the neuroimaging graduate student group, and all of you for being here today. Now I'm sure that you have already heard a lot about the role of the subcortical neuromodulatory systems in early Alzheimer's disease from Heidi Jacobs a few weeks ago. But today I would like to zoom in a little bit more on functional imaging of the locus coeruleus and discuss how this function of this nucleus is related to AD pathology and cognition in the early stages of the disease. And let me. Okay, here we go. All right, so here's the agenda for this talk. First, I'll make a brief introduction into Alzheimer's disease. Then I'll talk about the locus coeruleus and its role in AD. I'll then describe a methodological framework for functional neuroimaging of the locus coeruleus. And lastly, I'll present results from 3 studies demonstrating our findings about LC dysfunction in preclinical AD. So, as you may know, Alzheimer's disease is a neurodegenerative disorder causing the most common form of dementia observed in older people. It is characterized by a progressive decline in cognition, particularly memory and executive functioning. And but the disease is also characterized by a very long preclinical stage, during which the brain starts accumulating toxic proteins constituting the neuropathological hallmarks of the disease many years before the onset of cognitive symptoms. So this gives us an excellent opportunity to intervene before the onset of cognitive symptoms. Now, during this long preclinical stage of the disease, we have the accumulation of extracellular beta-amyloid plaques and intracellular neurofibrillary tangles of hyperphosphorylated tau. The accumulation of these proteins can lead to brain atrophy, which typically occurs at the later stage of the disease, and which is highly associated with cognitive decline. Now, accumulation of beta amyloid and tau follow two distinct patterns of progression. Using positron emission tomography, we see that tau pathology first appears in the entorhinal cortex, from where it progresses to the limbic system and then in the neocortex at the later stages of the disease. Beta amyloid pathology, on the other hand, first appears in the neocortex, then it progresses to the striatum and finally in the rest of the brain. And while this has been the classic view of AD pathology progression, more recently, autopsy data have shown convincing evidence that certain brainstem nuclei of the subcortical isodendritic core network, such as the locus coeruleus, the dorsal raphe, and others, accumulate AD-related tau pathology even earlier than in the cortex. For example, the locus coeruleus was found to accumulate tau even from early adulthood. And as shown in the figure on the right, at age 40, nearly everyone has tau in their LC. And by age 50, for 50% of people, tau has propagated from the locus coeruleus to the medial temporal cortex and the entorhinal cortex more specifically, which is quite frequently associated with cognitive symptoms, especially if beta amyloid is also present in the cortex. Okay, so and with this, I would like now to shift a little bit our focus on the locus coeruleus. So the locus coeruleus, or LC in short, sometimes I will refer to it as LC, is a tiny nucleus located in the dorsal pons of the brainstem near the fourth ventricle, and it consists of around 20,000 melanin pigmented cell bodies, which make it look like two little bluish dots during autopsy examinations. And because of this, the LC is frequently referred to as the blue spot. Now the LC, it is the main source of norepinephrine in the brain, which is released throughout the central nervous system via the LC's long and diffuse projections. And the neurons within the LC are organized in a way indicative of their projection destination. Rostral LC neurons, which is the upper part of the LC as you see in that image, and which is shown in green, supply norepinephrine to the cortex, to the neocortex, limbic system, and basal forebrain, including regions playing an important role in cognitive functioning, such as the amygdala and the hippocampus. Whereas caudal LC neurons, caudal LC neurons, here shown in yellow, project to the spinal cord and the cerebellum. Now, as the LC is the main source of norepinephrine in the brain, it is an important structure for autonomic control regulation, but it also plays an important role in norepinephrine-dependent cognitive processes, such as novelty detection, learning, and memory, but also in executive functioning. Which actually these processes are greatly affected by pathology early on during Alzheimer's disease. Now, due to the small size of the LC, perhaps the best way to examine it in vivo is using magnetic resonance imaging. Now, regarding structural imaging of the LC, Heidi Jacobs and her team, and I'm sure you heard a lot about this during her visit, have pioneered the development of LC sequences at the 7T, which show the LC as a group of voxels with a hyperintense signal, as you can see here, in these little hyperintense dots. That is used. So this hyperintense signal is used to quantify what we call LC integrity. Measuring LC integrity has been associated with AD pathology progression in the earliest stages, Braak stages, and it has also been associated with cortical atrophy and cognitive decline. However, measuring LC function is another interesting measure, as pathology-related changes in the LC function may precede structural alterations. Functional imaging of the LC using bold MRI is of course possible, but very challenging for a number of reasons. First, the LC has a very small size. It is approximately 14 millimeters long and 3 millimeters thick at its middle segment, which can lead to partial volume effects. Second, its elongated shape poses challenges in detecting activity, especially when adjusting for multiple comparisons using methods designed to detect spherical clusters of activated voxels. And lastly, LC imaging is particularly challenging due to the LC's proximity to the fourth ventricle, which exacerbates partial volume effects. This issue is especially pronounced when using special smoothing with special kernels to enhance, with spherical kernels to enhance the signal to noise ratio. And as depicted here with a yellow disc, this effect can contaminate the LC with signal from the fourth ventricle, as you can see here. But this is not the end of the story. Another source of noise in LC functional MRI is physiology. which, among other things, it causes brainstem motion due to cardiac pulsations, as I hope you'll be able to see in this video. So, as you can see here, you'll see a piston-like movement of the brainstem that is locked on cardiac pulses. So currently there are no dedicated sequences for LC functional imaging. So to improve the LC fMRI signal to noise ratio, perhaps the only thing we can do is to rely on dedicated LC preprocessing and bold fMRI signal modeling. Regarding LC bold fMRI preprocessing, in our work, we use standard pipelines. However, we found that tailoring two specific preprocessing steps to the LC can significantly enhance the LC bold fMRI signal to noise ratio. These two preprocessing steps, highlighted here with in yellow boxes, are weighted brainstem specific registration and spatial smoothing. And looking into each of these separately, our brainstem weighted registration is performed in 3 steps. The first step is functional to structural registration using boundary-based registration. This is a typical method that is used and implemented in many fMRI data processing toolboxes. And in the second step, we use an affine registration with the 12 degrees of freedom performed in 2 phases. In the first phase, structural images are registered to the MNI standard space using angular search and 12 degrees of freedom, as it was mentioned before. Then the resulting affine transformation obtained in this step is used as the initialization point for a second affine registration, this time with no angular search. However, in this second phase, we use weighting images, assigning higher weights within and around the brainstem to achieve the most accurate brainstem registration possible. And lastly, in the third step, we use nonlinear registration along with the optimized affine registration, a transformation that was obtained in phase 2 as the initialization point of this nonlinear registration in the third phase. Regarding spatial smoothing, instead of using spherical kernels, which were, as described before, are likely to introduce signal contamination from the fourth ventricle into the LC, we use ellipsoid kernels. These kernels are elongated along the z-axis, mimicking the natural shape of the LC in the same direction. Now, another critical analysis step for detecting LC activity is the use of LC-specific hemodynamic response functions. Now, as you may know, the bold fMRI signal is an indirect measure of underlying neuronal activity through a cascade effect that is known as the hemodynamic response to neuronal activation. This hemodynamic response is influenced by both neurovascular coupling mechanisms and vascular activation processes, which regulate blood oxygenation levels and in turn affect fluctuations in the bold fMRI signal. Quantitatively, the hemodynamic response can be described by the hemodynamic response function, here denoted by h of t, as you can see here, which I'm sure many of you have used extensively in your analysis. Now, given a bold fMRI signal measurement and the event timings of a task, the HRF can be estimated using various methods, but one particularly robust approach rooted in dynamical systems theory involves function expansions in terms of orthonormal basis functions. This function expansion technique enables robust HRF estimation even under conditions of lower SNR and relatively short data records, which is the case in most fMRI studies. The core idea of this technique is that the unknown finite hemodynamic response function can be expressed as a linear combination of orthonormal basis functions. But the choice of a basis set is crucial as it can influence these results. While any orthonormal basis set could be used, selecting an appropriate set can yield more parsimonious models. Okay, so in our work, we employ a variant of the Laguerre basis, the spherical Laguerre basis, which consists of orthonormal functions, the first a few of which resemble what we expect as a hemodynamic response function. The Laguerre functions depend on a parameter, a hyperparameter, here denoted by alpha, which controls the rate of their exponential decay. Therefore, by tuning this alpha parameter, we're able to obtain HRF estimates with faster or slower dynamics. So the slower dynamics extend longer in time, as you can see here. All right. I'll pause for a moment to ask if you have any questions. Otherwise, I'll just move on into how we can apply this for imaging LC function. All right, so with this methodological framework enhancing the LC Bolt fMRI signal to noise ratio, we aim to conduct a neuroimaging study to investigate in vivo how LC function changes in preclinical AD, but also its relationship to cognition. And specifically, we sought to confirm whether LC activity and LC cortical functional connectivity are involved in novelty detection, as actually it was demonstrated before in animal research, and to examine also their relationship with amyloid in clinically healthy older adults. Our second aim was to investigate the relationship between novelty-related LC activity, connectivity, and cognitive decline. To investigate this, we used data collected from 128 participants in the Harvard Aging Brain Study, which included baseline fMRI data collected during novelty detection, amyloid PET data, and longitudinal cognitive data. The amyloid data were collected using the carbon-11 PEEP tracer, and the cognitive data, cognition was evaluated using the preclinical AD cognitive score, which is denoted as PAC here. The participants were asked to perform a phase name association task during which they were instructed to remember phase name associations. The stimuli consist of unfamiliar faces that were shown either a single time as a novel phase or multiple times as a repeated phase. And the names were written below the phases, and a white fixation was alternated with a stimuli. We first conducted a voxel-wise analysis to assess whether the LC showed greater activation and connectivity during novelty compared to repetition. As shown in the image here on the left, we observed novelty-related activation within the LC, confirming the previous animal studies, but also in other regions, including the hippocampus, amygdala, insula, temporal fusiform cortex, and temporal occipital fusiform cortex. We also performed a voxel-wise psychophysiological interactions analysis and identified significant connectivity between the LC and both the amygdala and hippocampus, as shown here on the right. Lastly, incorporating beta-amyloid into our analysis did not alter this activity or connectivity patterns, and beta-amyloid did not show any significant relationship with LC activity or connectivity. We then examined the relationship between cognitive decline measured using the PAC and LC activity across different levels of beta-amyloid and observed a significant moderating effect of amyloid on the relationship between LC activity and cognitive decline. To visualize the directionality of this effect, we plotted the slope between cognitive decline and actually the slopes are shown here with an amyloid. So here we have PAC slopes and amyloid at 3 different levels of LC activity, and we observed the steeper cognitive decline when amyloid was elevated, particularly when LC activity was lower. And you see this here with a black line. Similarly, for novelty-related LC functional connectivity, we observed that lower novelty-related LC functional connectivity was associated with steeper cognitive decline in the bilateral hippocampus and parahippocampal gyri, particularly for those individuals with elevated amyloid levels. And as before, we also observe for LC connectivity that at elevated amyloid levels, steeper cognitive decline was associated with lower connectivity, LC connectivity. Now, in conclusion about this first part of the results, we observed no relationship between novelty-related brain activity or connectivity and amyloid, but we observed that lower novelty-related LC function was related to a steeper amyloid-related cognitive decline. In particular, greater LC activity and connectivity were related to slower cognitive decline, suggesting that proper LC functioning may contribute to resilience against AD-related pathology. All right, I'll pause for a moment to see if you have any question about this first study.
Hi, great talk so far. I did have a question that maybe you explained in the beginning, but I'm wondering if you guys tried this same analysis with any regions other than the LC. Like, is it that LC is particularly sensitive, which is what makes it worth imaging, given like how difficult it is to pre-process and like acquire signal from?
So let me go back to these maps. This was a whole voxel-wise analysis. It wasn't just in the LC. So
and it's just that the LC is the one that comes out as being really important.
One of the regions, but because we're really focused on showing the effect of LC dysfunction in early AD processes, we extracted the contrast values from the LC and we focused on those in this particular study.
Did you guys try any of the other regions to see if it could be like equally as sensitive?
I did it in the next study that I will present shortly.
Okay, awesome.
Okay.
Thanks so much.
All right. So moving on, the next part, the next investigation we performed, which was a follow-up actually on the study that I presented previously. As mentioned earlier, autopsy studies have shown that LC is one of the earliest regions affected by tau pathology, not amyloid. Previously, I showed results for the association between LC and amyloid, but we know from autopsy studies that actually it's tau pathology that is affecting very early during in AD the locus coeruleus. But also previous in vitro and animal studies suggested that pathological tau propagates through synaptically connected neurons with neuronal activity levels facilitating tau transmission. And this supports a potential relationship between LC activity and the progression of tau pathology. Also from human brain anatomy, as also demonstrated in the image here, using diffusion and MRI structural connectivity, we know that the locus coeruleus is densely connected with the entorhinal cortex. which is actually the first cortical region where tau pathology emerges. So here we have a very good setup to investigate whether there is progression. There is a correlation between the earliest region we know that is affected by tau pathology, that is the locus coeruleus, is in the brainstem. And as I will describe here in the AMS, the purpose here was to investigate in vivo the relationship between LC activity with cortical tau deposition using tau PET imaging and bold fMRI during encoding of phase name associations. And this perhaps will shed a little bit more light on the mechanisms of tau propagation from the brainstem to the cortex, even though we use cross-sectional data and we cannot really do causal analysis, but it will give us some information about the underlying mechanisms of how tau propagates from the brainstem to the cortex. And the second one was to investigate the relationship between novelty-related LC activity and memory decline and potential mediation by entorhinal tau deposition. To investigate this, we use data from the Harvard Aging Brain Study as before that were collected from 92 participants. But in addition to fMRI, amyloid PET, and longitudinal cognitive data, for these participants, we also had available tau PET data. Okay, tau PET. Let me find the pointer. All right, there we go. So tau PET, amyloid was measured as before with PEEP, and tau PET in this case was measured with fluortaucipir. For cognitive assessments for these participants, we had available a memory composite, an executive function composite, and PAC is the preclinical Alzheimer's disease composite score. So we first investigated brain activation during novelty versus repetition and observed significant activation in the LC, confirming the previous the results I showed you right before. So in addition to the LC, which now is shown in blue, we also found activation in other regions, but overall, these results here, which come from a different population that we initially used in the previous study, I mean, still we got significant similar regions showing significant activation during novelty. We then extracted the novelty versus repetition contrast values within the LC and investigated vertex-wise associations with cortical tau, which revealed significant associations in areas within the medial temporal cortex. And remember, the medial temporal cortex is the first cortical region where we detect tau tangles using PET imaging. And more specifically, regions including the entorhinal cortex and the inferior temporal cortex. Okay. Subsequently, we performed an ROI analysis and observed that lower novelty-related LC activity was associated with greater tau deposition in the entorhinal cortex. And again, the focus here was the entorhinal cortex, first of all, the LC, because we wanted to investigate the LC function as a mechanism of progression of tau from the brainstem to the cortex, and the entorhinal cortex is the first cortical region that tau is detected. That's why we focused on the entorhinal cortex here. I won't show images for other regions, but I'll say now that in addition to the LC, we also examined the association between the hippocampus, activity in the hippocampus and the amygdala, in predicting entorhinal tau, and we found no significant association. So this was very specific to the locus coeruleus. When examining the direct associations between novelty-related LC activity and cognition across different domains, we observed a stronger relationship between lower LC activity and memory decline, but not with executive function or the preclinical Alzheimer's disease composite score. So these associations in terms of cognition were very specific in the memory domain. Now, given the observed relationship between novelty-related LC activity and cortical tau, and the close coupling between early cortical tau pathology and memory decline that has been reported in the literature, we sought to determine whether the relationship between LC activity and memory is mediated by tau deposition in the entorhinal cortex. To achieve this, we conducted a mediation analysis with the results shown here on the right panel, and we found significant tau mediation effects, suggesting that the impact of LC activity on memory decline is partially influenced by tau levels in the entorhinal cortex. Now, our data were cross-sectional. We cannot infer any causality with this, but based on what we know from autopsy studies here, the way we form this mediation analysis is based on the observations and reports in the literature suggesting that brainstem regions, including the LC, are the earliest regions where we can have AD pathology. This may affect LC function, which then plays a role in the progression of tau pathology in the cortex. And this is the entorhinal cortex. It's the first cortical region where tau is detected. And because of the progression of the disease to the cortex, then we also have a decline in cognition. So this mediation model is absolutely based on our hypothetical evolution of the disease. I just wanted to make sure that we cannot conclude anything about causality. But still, we see some hypothetical progression of the disease based on what we know. We confirmed our hypothesis based on what we know in the literature. But we also conducted moderated mediation analysis to examine whether the mediating effect of tau on the relationship between LC activity and memory differs in the presence of amyloid. And as shown here on the right panel, the analysis revealed significant differences in the mediation effect of tau between amyloid positives versus negatives. And then to further examine the effect of amyloid on each component, each arm of the mediation model, we conducted Johnson-Niemann floodlight analysis. This analysis revealed the association between novelty-related LC function and tau deposition in the entorhinal cortex, which you see here, becomes significant at lower amyloid levels compared to the association between tau and memory decline, which again suggests that LC dysfunction and tau propagation may precede AD-related memory decline, because the level of amyloid where these associations between LC dysfunction and entorhinal cortex become significant are at lower levels of amyloid as compared to when cognition, the association between tau and cognition becomes significant. All right, so in conclusion, in this second part, in this follow-up work, we found that lower novelty-related LC activity was related to greater cortical tau deposition in regions showing early AD-related vulnerability. And I'm talking about the entorhinal cortex here. Again, it's the first cortical region. These results suggest that LC dysfunction may be linked to tau pathology progression, which has further implications on mechanisms underlying tau spreading from the brainstem to the cortex during the earliest stages of the disease. In addition, we observed that while the relationship between cortical tau and memory decline was more pronounced at higher levels of amyloid, in agreement with previous research suggesting a synergy between cortical amyloid and tau pathology on cognitive decline, the association between LC activity and cortical tau was more pronounced at much lower and potentially subthreshold amyloid levels, as I mentioned before, which again highlights the early involvement of LC in cortical tau pathology. All right, so this concludes the second part of the results that I wanted to present, and I'll pause again for a moment to see if you have any questions at this point. All right, so very briefly, I'll go through the last section. So these are some of our latest findings on the relationship between LC function and fluid biomarkers of AD-related tau phosphorylation. Now, as you may know, tau PET imaging detects aggregated forms of tau, okay, aggregated forms, which represent more mature stages, as you can see here, more mature stages of tau pathology. However, recently developed fluid biomarkers of tau hyperphosphorylation, in addition to tau aggregates, may also reflect earlier stages of tau pathology. Okay, so given the early involvement of the LC in AD, this study aimed to investigate the relationship between LC dysfunction and plasma biomarkers of tau phosphorylation. Also, animal studies using DSP4, which is an acute neurotoxin, demonstrated that LC depletion, so complete LC knockout, exacerbates astrogliosis and AD pathology in rodents. And in addition to this, in vivo imaging preclinical AD patients, so this is human studies, further suggested that astrocyte reactivity may be an important upstream event linking early AD-related pathophysiological events, such as cortical deposition of amyloid plaques to increases in plasma tau. But from neuropathologic studies, we know that cortical deposition of amyloid plaques is not the earliest pathophysiological event in AD. In fact, we know that tau accumulation in the LC occurs before that. So the role of astrocyte reactivity in the relationship between LC dysfunction and AD-related tau pathology in preclinical AD remains elusive. And therefore, in this study, the main objectives were to investigate in vivo the relationship between novelty-related LC function and levels of hyperphosphorylated tau in the plasma, and whether this relationship is potentiated by astrocyte reactivity. So in this study, we used data collected at the 70 from 79 participants in a lifespan study conducted in the Netherlands. And the data set included baseline fMRI data collected during the execution of an affective memory task, blood biomarkers of tau hyperphosphorylation, including p-tau 217, 231, and 181, and GFAP, which is glial fibrillary acidic protein, which is a measure of astrogliosis. The task comprised of emotional images selected from the International Affective Picture System, and they were equally divided into high arousing with negative valence images and low arousing with neutral valence images. Since we had no prior hypothesis about which p-tau species might best reflect LC activity, we computed a plasma p-tau composite score derived from the 3 tau biomarkers included in the study. And using this p-tau composite, we observed that lower LC activity during high versus low arousal was associated with greater p-tau levels in a cluster of voxels within the right LC. But this p-tau measure we will use here was a composite made of the 3 biomarkers that we had. So then we looked more specifically into which we examined the associations between LC activity and each biomarker separately, and found that lower LC activity was associated with higher p-tau 217 and 231, but not with p-tau 181. And then we investigated the moderating effects of GFAP, astrogliosis, on the relationship between LC dysfunction and tau phosphorylation. And we found significant moderating effects by astrogliosis on that association, except for the association between LC dysfunction and p-tau 231. All right, but since p-tau 217 was found to be associated with LC activity more consistently compared to the other biomarkers, we selected it as a representative plasma p-tau biomarker for subsequent analysis, including LC cortical functional connectivity. And we repeated the same analysis using LC functional connectivity. And with this, I mean the connectivity between the LC and the cortex. And we found that lower LC connectivity was related to higher levels of p-tau 217 in the blood. And again, this association was moderated by GFAP, by astrogliosis, very similar to what we've seen for LC activity. Lastly, we investigated the relationship between LC activity and connectivity, tau phosphorylation in the blood, astrogliosis, and cognitive decline. And we observed no significant association between LC activity and cognition. However, we observed that LC functional connectivity was related to plaque decline, to cognitive decline, when both p-tau 217 and GFAP were elevated. Okay, so in conclusion, here we found that astrogliosis exacerbated the effects of locus coeruleus dysfunction on tau. And we also found that LC dysfunction, as reflected by decreases in LC cortical functional connectivity, was associated with cognitive decline, particularly in individuals with elevated astrocyte reactivity and plasma tau. And this suggested that the synergy between AD pathology and astrogliosis may compromise brain integration, which is very important in cognition, as demonstrated in previous animal and human imaging studies. And really quick, an overall conclusion about this work. I hope I demonstrated to you convincing results suggesting that LC dysfunction may represent an early pathophysiological event in Alzheimer's disease. And in particular, maintaining proper LC functioning may be beneficial in delaying AD-related cognitive decline. These findings, we think, they have also important implications for the design of early interventions. And for example, and I'm sure you've heard more about this from Heidi Jacobs a few weeks ago, the Jacobs lab here at MGH is currently conducting a clinical trial in which participants receive tiny electric pulses in their earlobes designed to increase LC activity via transcutaneous vagus nerve stimulation. So whether TVNS has a beneficial effect on cognition, on memory, and whether it can promote resilience is something we're hoping to learn in the next couple of years. And this concludes my presentation. And with this, I wanted to thank all of our participants, my colleagues at the Blue Spot Lab, my mentor, Heidi Jacobs, who had the general supervision of this work, and also you for your attention. And I will be very happy to take any questions of yours. Thank you very much for your attention.
Yeah, hi. Fascinating work. Just a bit of a broad question, but like,
Sure.
how are you finding things like motion to be impacting the way you are processing the LC signal?
Yeah. Well, cat motion, it's an avoidable enemy, of course. Yeah, I think, let me go back to that slide. So undoubtedly it affects it, but what we do, there is a box here. I didn't talk about much about it, but first of all, we performed nuisance regression, including 24 parameters derived from the 6 motion parameters we get from motion realignment. So in addition to this, we also performed ICI aroma, which is an independent component analysis based technique. It's a classifier basically, which has been developed specifically to account for motion in the data. We've been using this. We also compared this with other existing classifiers that potentially detect motion artifacts like FIX, again, ICA-based classifiers, which actually we use labels trained, derived from our own data. But we found ICA aroma performing the best. So we do this as a measure to account for motion. If it really removes all existing motion artifacts, I don't know, and I doubt. But I think it's, at least we're accounting for that to a good possible way we can do this. In addition to this, I should also mention that we include regressors, anatomical components derived from the fourth ventricle. Okay, so motion, because this region, the fourth ventricle, is really close to the LC, it should be affected in a similar way. It should be affected in a similar way, by motion in a similar way as the LC. Now, the fourth ventricle is full of fluid. Fluids have different, properties in motion. I mean, with respect to motion, they probably move more than the more solid tissue. But we also include regressors derived from the fourth ventricle, and hoping that in addition to physiology, these regressors also inform information specific to the region around, motion-related information specific to the region around the LC. So yeah, this is what we do.
Yeah, it was a really great talk. I really enjoyed it. I did have one more question just from your third study. So I don't know a lot about tau or a lot of like AD biomarkers. But I know in your last one you were saying that like 217 was the one that seemed to be the most consistent and showing this like low activity to high p-tau relationship. But I know you tested 3. Do you guys have an idea of maybe why that one came out and not the other 2, of like how their roles might be different?
I mean, I should say, well, about the specific biology of each, the answer is no. I don't have a specific biological explanation why that one.
Sure.
I should say that, first of all, from what we know about blood biomarkers, is that they're all correlated. To some extent, they convey similar information, and that's why at the beginning we started with a composite score. Derived here, we started from that, and then we looked separately each one. But in general, yeah, so we know that p-tau 231 is more sensitive, perhaps is more sensitive in detecting very early stages, like transitioning from like BRCA1 stage to BRCA2. On the other hand, p-tau 217 is more robust as a biomarker, so it's more reliable. We found, in addition to p-tau 217, we also found for activity, we found some significant associations also with p-tau 231. P-tau, where is it? P-tau 181, on the other hand, this correlates a lot with age and also with amyloid. So it may not be as specific to early tau formations as the other two, especially p-tau 231. But overall, they're all very correlated. the one that was more consistently found activated in all our analysis, including both LC activity and LC connectivity with the cortex, in our study was p-tau 217. Now,
Yeah, which you had said
if,
seems to be a little more reliable as a blood marker, so that can make sense.
if it tells that, I shouldn't, I'm not feeling very strong about it.
Yeah.
But the way I interpret these results is that there is an indication of LC dysfunction plays a role in early tau phosphorylation processes, which are indicative of AD. This is the way I would like to present that, and
Mhm.
of course, time will show. We learn new things about biomarkers every day. So yeah, what is the specific biology behind them, and what is the relation of this biology to the LC? It's something that I don't know, and I'm
Yeah, it sounds
happy.
like maybe everyone is still
Yeah.
kind of figuring it out.
Right.
Okay, last call. Any final questions? Okay, let's thank our speaker one more time.
Thank you, thank you everyone, guys, and thank you also for the invitation.
Yeah, thank you so much. Have a nice day.